The next presenter will be Dr. Go from Utah. Her talk is about the genotyping in Malaysian's banana cultivars using a sequence characterized amplified regions system, SCAR system. So please welcome Dr. Go to share her talks on the banana classification. Thank you.
Okay, thank you, Dr. Teo. So I hope you can hear me.
Yes, yes.
Okay, thank you. So today I'm going to share with you all kind of a small preliminary results of collaborative work between United Plantation and me. So it is about genotyping the banana cultivars using a SCAR marker system developed previously. So we are testing out the existing marker system to identify cultivars in Malaysia. So all right. So in my introduction, I'm going to follow the conventional AB system. Okay. So because this is prepared before the revolutionizing talk by Dato Sri. So but anyway, since until now, a lot of people are still using this system. So let me introduce a little bit on the AB system so that we all know what is happening. So this banana refers to the Musa genus, and it belongs to the Musa CE family, the order of gingiberries. So the cultivated bananas and plantains are seedless, and the fruitings are mainly 2 parthenocarpy, as mentioned and explained in very detail by Dato Sri. So these AB genomes, the nomenclature are already explained by Dato Sri just now. So basically, generally, the general belief is that the A genome arose from this acuminata, and the B genome arose from balbisiana. But the edible bananas and plantain, there is no clear-cut botanical distinction. In general, the so-called banana is the sweet dessert banana, and the plantain is the cooking banana. Most of them are triploids, some of them diploids and tetraploids. The basic chromosome number in general for banana is 11. So this is the list of the banana cultivars in Malaysia, some of them. The domestication history. General belief is that acuminata is centered in Southeast Asia. So it can be divided into a number of subspecies. For example, Malacensis, the micro kappa, errans, Zebrina, Banksia, and so on. So, but the balbisiana mainly distributed in further north, which is subtropical mainland Asia. So the domestication mechanism involves wild diploids, edible diploids, and then it comes to the edible polyploids. It was the general speculation is that the spontaneously occurred mutants in the wild were collected and then selected and maintained by people, and then and of course because there are 2 species here and multiple subspecies here, there were crossbreeding mechanisms involved here, either interspecies or intraspecies. So learning from this kind of domestication mechanism, we can expect the banana to have diverse cultivars, diverse type of bananas, because multiple ancestral gene pools. But within each cultivar, they should be homogeneous, they should be uniform because of the nature of vegetative propagation. So this is the map by Perrier. Yeah, so Perrier's, later I'll come more about this Perrier's work. Perrier basically genotyped the so-called subspecies and found that the subspecies are the molecular results corroborates the natural distribution of the subspecies. So, genotyping of the acuminata and balbisiana, there are a lot, quite a lot of phylogenetic work. I think later on, one of our plenary speakers will also talk about that. So I can only list a few examples here. Basically, there are many genotyping methods, right? So some are profiling methods, DNA fingerprinting methods, for example, RFLP, AFLP. The more commonly used for genotyping methods, more popular one is the microsatellite, and many other like retrotransposon done by this Dr. Teo. Okay, so there are many genotyping methods. So these methods were developed mainly to distinguish cultivars or to match the closest possible wild origins, or to assess the diversity of the acuminata subspecies, or maybe just Musa species in general. So most of these are regional efforts, emphasize on the cultivars of interest, which is more localized. Okay, and then we also find that this balbisiana, the work on balbisiana is relatively less. I think perhaps it is the like what was described by Dr. Sri just now, because people are really confused about what is true balbisiana. All right. So if following the concepts, Dr. Sri's concept about known crossbreeding between the banana species or banana subspecies, we could probably speculate, it's just a speculation, speculate that the existence of so many subspecies or so many different species or different groups, whatever taxonomy level you want to call it, existence of these different bananas, wild bananas, should be because of the they could be the residues of the wild progenitors, which are already different. Otherwise, we won't see so many different types of banana. So I still believe at one point, maybe already extinct, but at one point there should be a pro banana which is still able to reproduce sexually. All right, so that is just some speculations. So okay, so this work is actually following the scar marker system developed by this research group by Boonruangrad, okay, in 2009. They sequence the nuclear ETS rDNA region, and then they developed subspecies specific markers to trying to make a rapid method, okay, to detect the banana subspecies, as well as the cultivars which are derived from those subspecies. Okay, so basically it is the single nucleotide polymorphism that are specific to each subspecies and being identified, and then being used to design the subspecies specific PCR primers. So in their publication, 53 banana samples with the known identity, both wild and cultivated ones, are tested. So the subspecies of acuminata were grouped into A1, A2, A3, A4A, and A4B. Okay, A4A is this group, this huge group here, which covers the Banksii, Irans, and the Microcarpa. Whereas A4B is not illustrated here, but A4B is Malacensis. Okay. So the objective, we have a very simple direct forward objective to determine the effectiveness of this marker system on the selected banana cultivars to elucidate the subspecies genomic composition. So indirectly, we assess the potential of this marker system to be developed into a low cost and rapid, a very convenient genotyping method for the cultivars of interest. So we tested this marker on pisang Jarum Ali, probably the what lady's fingers banana variety just now, and the Berangan, which are obtained from United Plantation. So these are micropropagated materials actually. So total DNA was extracted, and then we apply the primers developed by Boonruangrad. This is so imagine this is the piece of DNA of the ETS region. So the forward primer was designed on the site 280, and the reverse primer were designed at the site 801. So in between, there are a number of forward primers, okay, designed at the point, at the snip point, which can differentiate different subspecies. Okay, so in the end, the presence or absence of the band will indicate the presence or absence of the A genome of each subspecies. Okay, so we tested all these primers to look at the band profiles. Plus, we also sequenced the amplicon, right? So the results tested on only 2 cultivars, okay, preliminary. So Berangan supposed to be triploid. However, we can see there are 5 bands here. All 5 are present. So A1 indicating zebrina, A2 melacensis, A3 here Bermenica or simonsis, then A4 all this. So all of these are possible. Okay, on the other hand, Jarum Ali diploid, we found A1, A2, A4, 4A, and 4B. So there are also many possibilities. So then we feel not very satisfied with this one, because how can the the triploid and diploid holding so many different subspecies origin? So then we examine the sequence. Okay, direct sequencing was done, and we compare the Berangan, the Berangan sample that we had. Then we fit the primer sequence onto the sequence we obtained. Then we plus another so-called Musa AAA from GenBank, but it is not stated what is that exactly. So, and the first line is the site number. So the site number starts at 280 and it ends around 800. So we found that SNP sites that are specific to the subspecies are circled here. Okay, according to mapping to the information given in Boonruangrad's publication, these are the sites which are supposed to be specific to the subspecies. All right. Somehow, we found that this one on the first marker, on the first primer, and this one on the 4B primers, only these 2 are matching the primers. Okay. And on the other hand, there are a few mismatching here. Okay, these are the mismatch. Okay, so what we learned from here is that the profile, okay, by merely looking at the DNA binding profile, we can't really determine. Okay, and from the DNA sequence data, we can see that they are mismatching, meaning that the primers are not very specific. Okay, this marker system based on the DNA bands is prone to unspecific priming, and so the results is less reliable. Okay, so the DNA sequence data should be more reliable. It shows correct matching. So for Berangan just now, we we find that it is matching the A1 Zebrina and A4B Malacensis. Hopefully it makes sense. Okay, so yeah, so we think that direct sequencing for this ATS rDNA region is showing much promise. So, but of course, we are going to test more cultivars and including more replicates. So to confirm to the sensitivity and the specificity of the SNPs that could indicate the subspecies or species. So yeah, I don't have big results like Dr. Teo, okay, but some afterthoughts of this. I'm of the opinion that rapid genotyping methods for the cultivars of interest can actually be directly obtained or established based on the DNA available, data available, okay, to achieve specific goals for the stakeholders or smallholders, okay. So in case of limited resources, okay, now everyone is, you know, knows that funding is very limited, okay. So we have very limited resource, manpower, and funding. So the SNP markers and microsatellite markers developed by Boonruangrad and Perriers are worth trying. And these markers actually can provide some indications of the possible wild progenitors. And the conventional multi-gene analysis approach generates DNA data that can be conveniently shared and compared and reanalyzed by different research groups. Okay, for example, you see Boonruangrad's DNA DNA fragment once sequenced and deposited into gene bank. Anyone can take the sequence from gene bank and then get additional data and then reanalyze. That is convenient compared to the high throughput sequencing. And the genotype morphotype records can then be established and expanded, okay, by researchers of different locations. These are Perrier's research. Perrier's use actually, I think, if not mistaken, Perrier used 22 microsat markers. So in the end, they came up with this distribution map. I mean, distribution, they call it a factorial analysis. So it shows the distribution of the varieties in their experimental design. So this Zebrina is here, which is far from the bank CI, and then Malacensis in blue, and this Bremica semia in brown. Okay, Microcappa is to them is too diverse to be considered as a natural group, and Truncata also quite diverse. That is what they found out. So, but they used 22 microsatellite markers. Okay, so of course, to develop a more universal and robust solution, the high throughput approach, which is those that are using NGS, next generation sequencing, should be pursued. I think Dr. Teo showed us a good example just now. So challenges are high initiation cost in terms of technical and manpower or expertise. And then the whole genome sequencing is less achievable for the organisms with huge and complicated genomes like banana. Then now for population genomic studies, people like to use reduced representation genomic sequencing, but this data may not be directly comparable with or may not be readily reanalyzed by other research groups unless they use exactly the same molecular methods, right? So the high throughput approaches can aim to develop a set of informative markers by identifying the genome-wide SNPs or point mutation and microsatellites or whatever repeats for the Southeast Asian subspecies. And I think this genotyping will only be meaningful when the morphotyping is also, being set alongside. And more markers can be used for genotyping and population and evolutionary studies. So of course, last but not least, the regional efforts or global efforts should be should also be pursued. And this should involve both breeders and forest botanists, so that we can improve the genotyping system, as well as improve our understanding on the evolution and domestication of bananas. So I think that is all from me. Thank you very much, and these are my reference list in case you are interested. Okay, thank you.
Right. Thank you, Dr. Goh, for sharing your interesting finding on using the scar marker. So I hope that we can actually use this scar marker, especially on the Ambon accession coming from Dato Sri Lim. So I mean, it's better to have a different marker system to sort out the taxonomy of banana, especially if you want to declassify the the acuminata and balbisiana. Yeah. So I hope that in future we can work together to sort this thing out, at least to know to apply for grant and to work together and to get this done. Yeah. So I think we have already ended our time. Yes, I think everybody will go back to the poster viewing section after that. But can we actually stay for a while to discuss a bit?
Can I contribute one or two things?
Yes.
I think we can maybe we can pass it to the MC first, and
yes,
those who want to stay back and discuss can do so.
All right. Okay. Thank you. Yeah. Now pass back to MC. Thank you. Kiva.
Thank you, Dr. Teo. Thank you to all the speakers. Right now, we have a short break for lunch and also poster viewing, and the next session will commence exactly at 2 p.m. But in order to facilitate a smooth transition into breakout rooms and all of this, so please be at the main room at 1:50 p.m. To participants who want to stay behind and listen to the discussion, please feel free to do so right now. Thank you.
All right. Thank you. Yes.